Hello students, welcome to EPG Pathshala. Myself Dr. Sheetal Shirodkar. I am working as an assistant professor in Amity Institute of Biotechnology, Amity University, Noida. Today I will be presenting a class on the topic Applications of Biotechnology from the paper Molecular Biology, Genetic Engineering and Biotechnology. The objectives of our study will include introduction to biotechnology, explain about genetic engineering as an important tool in biotechnology, explain about applications of biotechnology in varied fields with examples, and explain about ethical issues associated with biotechnology. Coming to the concept map, biotechnology has applications in varied fields including plant biotechnology, animal biotechnology, food biotechnology, environmental biotechnology, pharmaceuticals, industries as well as forensic sciences. Biotechnology is defined as a set of tools that uses living organisms or parts of organisms to make or modify a product, improve plants, trees or animals, or develop a microorganism for specific use. Some of the biotechnological applications that we will be discussing today includes its use in plant biotechnology, animal biotechnology, pharmaceutical, environmental, food, industrial and forensic sciences. Genetic engineering. Genetic engineering is an important biotechnological tool. It makes use of recombinant DNA technology for inserting new genes or removing pre-existing genes, resulting in generation of a genetically modified organism. Figure 1 explains about the basic steps in genetic engineering to produce a transgenic plant or a genetically modified plant. There are 5 steps involved in the process. First, the plasmid DNA is isolated from the bacteria. Plasmids are extrachromosomal DNA material that replicate independently of the chromosomal DNA. It contains multiple cloning sites which allow easy cloning of a foreign DNA to be cloned and selection markers that allow selection of recombinant plasmid during the cloning process as well as selection of the transformed plant cells. In the next step, the recombinant plasmid carrying the foreign gene of interest is transferred into the plant cell using a microprojectile gun or by making use of agrobacterium mediated gene transfer. The transformed plant cells are transferred aseptically to a suitable culture medium so that the cells divide. The dividing cell mass is then transferred to a suitable culture medium supplemented with hormones that allows plant regeneration. Once the plants are hardened, they are adjusted to the natural environment. The plants thus produced are known as transgenic plants. Biotechnology is defined as a set of tools that uses living organisms or parts of organisms to make or modify a product, improve plants, trees or animals, or develop microorganisms for specific uses. Traditionally, improvement in plants and animals were done through breeding or hybridization methods which were time consuming, laborious and may combine desirable traits with undesirable ones. In 1970s, with the development in the field of molecular biology, scientists were able to manipulate genes which are basic physical and functional unit of heredity and is made up of DNA. This technology is called as genetic engineering and is an important tool that allowed transfer of genes to any plant, animal or microbe, resulting in generation of genetically modified organisms. Thus, genetic engineering has allowed desire manipulation in a plant, animal or microbe in a more rapid manner compared to the traditional breeding methods. Biotechnology has made an impact on varied fields like agriculture, medicine, genetically modified organisms, poultry, etc., to name a few. In the following section, we'll discuss genetic engineering as an important tool in biotechnology and discuss about its applications in varied fields. Figure 10. Now we'll discuss about monoclonal antibodies, their production and few of its applications. A bacterial cell possesses a number of epitopes on the cell surface, and the injection of such a bacterial cell into the host system would result in development of a variety of B cells, each interacting with a different epitope. Thus, the resulting antiserum will contain a mixture of antibodies specific to the various epitopes. Such a response is known as a polyclonal response, and the antibodies produced are known as polyclonal antibodies. However, for experimental, therapeutic and diagnostic purposes, monoclonal antibodies are required, which are antibodies produced by a single clone of B cell capable of identifying a single epitope. Commercial production of monoclonal antibodies is done by hybridoma technique, which was introduced by Kohler and Melston in 1975. In this technique, antibody producing B cell is immortalized by fusion with the myeloma cell line. The resulting hybrids produced large amount of monoclonal antibodies to be used for various purposes. Some of the common application of monoclonal antibodies are in diagnostic and therapeutics. In diagnosis, small amount of drugs, toxin, proteins and hormones can be detected from various body fluids such as serum, blood, urine, etc. by making use of monoclonal antibodies. For therapeutic applications, monoclonal antibodies can often be modified by conjugating it with a toxin or a radioisotope. Such monoclonal antibodies specific against cancer antigens can be used to specifically attack and kill cancer cells through the action of the conjugated toxin or radioactivity. Plant biotechnology. We'll now discuss about the applications of plant biotechnology. Plant biotechnology is a field of biotech that results in improvement of plant health, quality and productivity. Traditionally, plant breeding methods were practiced to select for beneficial characters of the plants. However, the process requires selection for several generations so that the plants with a set of desired characters can be obtained. In the last few years, plant breeders have been able to precisely incorporate desired characters in the plants by making use of genetic engineering methods. Some of the plant biotechnological applications which will be discussed includes micropropagation, production of golden rice, pest-resistant transgenic plants expressing the Bacillus thuringiensis genes, and the flavor Savor tomatoes. Micropropagation. Normal vegetative propagation of plant is slow and seasonal. Hence, micropropagation technique is used for large-scale multiplication of plants. The technique makes use of small tissues of plants like leaf, stems, etc. as explants, which are surface sterilized and cultured in a suitable plant tissue culture medium supplemented with plant hormones. The regenerated plantlets are hardened in a greenhouse and ultimately planted in soil as a normal plant. Micropropagation allows rapid multiplication irrespective of seasonal variation. It allows producing large number of plantlets in a small laboratory space. It also allows multiplication of sexually sterile hybrids and production of disease-free plants. Use of recombinant DNA technology has allowed us to improve the food nutritional values. This can be a very important source of nutrients for the poor population in developing countries who suffer from malnutrition and nutrient deficiency. Golden rice has been one of the important applications of genetic engineering in plant biotechnology. The expression of the beta-carotene gene imparts a golden yellow color to the grain. The beta-carotene is converted to vitamin A after digestion, thus supplying the necessary vitamin A requirements. Other genetic engineering implementations in agricultural biotechnology includes increased protein content in soya bean, amino acid content in beans, etc. Crop losses because of insect pest are huge, resulting in considerable financial loss to the farmers and decrease in crop productivity. Farmers typically make use of tons of chemical fertilizers to combat the insect pest. However, consumers are not willing to eat such food because of potential health hazards. Researchers are now able to generate transgenic crops such as corn, cotton, potato that express the Bacillus thuringiensis toxin gene, the Bt gene. When an insect consumes such a transgenic crop plant, they also consume the toxin. The toxin becomes active when it reaches the alkaline environment of the insect's digestive tract. The active form of the toxin then inserts itself into the gut cell membrane, resulting in formation of pores and breakdown of the gut wall, allowing spores and the gut bacteria to enter the body. The insect ultimately dies of septicemia as the gut spores and the microbes proliferate in the body. Growing such GM plants can eliminate the use of chemical pesticides and reduce the production cost. Improvement in shelf life of the fruits is another important application of agricultural biotechnology in which the ripening process is slowed. This allows long distance transport of fruits without damaging it and also increases its shelf life and flavor. Fruit ripening process involves degradation of the pectin component of the cell wall because of the activity of enzyme polygalactouronase. Transgenic tomatoes were produced based on antisense technology in which the cDNA complementary to the polygalactouronase gene was expressed, thus preventing expression of the polygalactouronase gene. Animal biotechnology. Animal biotechnology makes use of genetic engineering and other biotechnological tools for improving animal health and productivity of the animal-based food products like meat, milk, eggs, etc. Its applications include animal cloning, improving animal health, and improving productivity of animal-based food products like meat, milk, eggs, etc. Some of the applications that we'll be discussing today includes animal cloning and generation of knockout mice. Animal cloning. In cloning process, a somatic cell containing nucleus is fused with an enucleated egg cell by application of an electric shock. The fused egg is then cultured in an appropriate nutrient medium till it develops into an embryo. The embryo is then transferred and transplanted into a surrogate mother. The resulting progeny will have the exact genetic complement to that of the somatic cell donor and is known as the clone. The sheep Dolly was the first ever mammalian clone that had been made through this technique in 1997. Dolly was able to mate and produce offsprings in a normal way, suggesting that such cloned animals are reproductively fertile. The advantages of animal cloning is that a particular genotype that has been proven to be well adapted in a particular environment can be maintained indefinitely without any genetic changes that happen during reproduction. Generation of knockout mice. Knockout mice has applications in research to study the role of a particular gene or to study the effect of a particular drug on the gene. In this, a specific gene is disrupted or removed such that it is not expressed. In this method, first the insert gene with an antibiotic marker is cloned into a vector. The plasmid DNA is then incubated with the embryonic stem cells. Three kinds of products result from the incorporation of the plasmid, and one of which is the knockout. The modified embryonic stem cells are then introduced into the normal embryo. The embryo is implanted in the incubator mother. The offspring is a chimera. It may take several generations of crossbreeding it may take several generations of crossbreeding to produce animals that are complete knockouts. Pharmaceutical biotechnology. Pharmaceutical biotechnology makes use of biological systems like plants, animals, or microbial cells and tissues for manufacture of pharmaceutically important products. These include many enzymes, proteins, hormones, etc. Traditionally, these pharmaceutical products were directly extracted or obtained from their source material. Some of the source material were dangerous and undesirable. For example, the human chorionic gonadotropin hormone was extracted from urine of pregnant women. However, with the advent of biotechnology and genetic engineering, the production of these pharmaceutically important products was done by cloning the required gene sequence in an appropriate biological system like plants, animals, or microbes, and the products were harvested from the transformed cells in culture. Some of the pharmaceutically important products that we'll be discussing includes therapeutic hormone insulin, therapeutic enzymes, anticoagulant hirudin, monoclonal antibodies, and vaccines. Insulin. Figure 9. Therapeutic hormone insulin is a peptide hormone which is produced by the beta cells of pancreas. It is made up of two short polypeptide chains, A and B, linked together by disulfide bonds. It is required to control and regulate the blood glucose levels by stimulating its uptake into the cells. Lack of sufficient insulin production by body leads to development of disease condition known as insulin-dependent diabetes mellitus or type 1 diabetes. Long-term side effects of untreated diabetes include cardiovascular diseases, stroke, kidney dysfunction, foot ulcers, and damage to eyes. The blood glucose levels in such patients can be controlled by parenteral administration of such insulin preparations. Traditionally, insulin hormone was prepared by extraction from pancreatic tissue of slaughterhouse animals, followed by its purification by chromatographic methods. However, such animal-derived insulin had side effects such as immunogenicity in humans and shortage of insulin because of limited number of slaughterhouse animals. These limitations paved the way to use of recombinant DNA technology based production of insulin. Eli Lilly and Company were the first pharmaceutical companies to produce recombinant human insulin by cloning and expressing the human proinsulin gene in E. coli. The expressed proinsulin peptide was purified and cleaved to obtain the final active insulin. Vaccines. Vaccines are biological preparations of microorganisms which are given with the aim to provide active immunity against a particular disease. Different types of vaccines are currently in use in market. The kill vaccines are inactive preparations of microorganisms made through treatment with chemicals such as formaldehyde. They provide limited immunity and repeated booster doses are required for long-lasting immunity. Live attenuated vaccines, on the other hand, are live preparations of microorganisms which are made non-pathogenic or avirulent through repeated subculture on a foreign host such as cells in culture. Live attenuated vaccines provide long-lasting immunity since the cells are dividing. However, they may sometimes revert to its infectious forms and cause disease. DNA vaccines are another important breakthrough in the production of vaccines in which the antigenic protein gene are isolated and cloned into an appropriate vector. These are then directly injected into the muscle or the skin cells of the person to be immunized. Expression of the plasmid-borne antigenic protein takes place directly in the cells of the immunized organism, providing long-lasting immunity. One of the major drawbacks with the use of DNA vaccines is the possibility of integration of the plasmid into the nuclear genes, leading to new mutations that may be lethal or have side effects. These vaccines are still in experimental stages and clinical trials are not yet been done in humans because of the ethical issues associated with the use of DNA vaccines. Environmental biotechnology. Environmental biotechnology deals with use of biotechnological tools to resolve issues related to environment contamination and preservation. Intervention of human activities leads to generation of waste from different industries. Agricultural runoff as well as domestic waste are a few causes of environmental contamination. These contaminants include pesticides, herbicides, fungicides, heavy metals, solvents, petrochemicals, etc. These environmental pollutants affects growth of plants, animals and microbes. Some of these contaminants like pesticides and heavy metals are also toxic to humans and animal health, leading to diseases and even death. Thus, restoration of environmental health is important to protect the living system from all the ill effects of contaminants and to improve the quality of life. Some of the important applications of environmental biotechnology that we will be discussing includes pesticide degradation, mushroom cultivation and biosensors for monitoring environmental contaminants. Pesticide degradation. Pesticides are chemicals or compounds intended for preventing growth or repelling different forms of pests including weeds, insects, rodents, mites, etc. However, the main concern with the use of pesticides is their persistent nature in the environment and their effect on non-target organisms including humans, animals, plants and microbes. Several laboratories worldwide are working on isolation and characterization of microbes capable of degrading several groups of pesticides. For example, organophosphate pesticides and nitroaromatic compounds. Some microorganisms capable of degradation of these group of compounds include arthrobacters, Pseudomonas species, etc. These microorganisms express an enzyme named organophosphorus hydrolase encoded by the OPD gene. Through the use of recombinant DNA technology, the OPD gene has been cloned and expressed in many bacterial systems such as E. coli and Pseudomonas. The biodegradation capacities of such genetically modified bacteria can be further improved by metabolic engineering and manipulation of regulatory networks. These GM bacteria can be exploited for bioremediation of contaminated environments as well as degradation of industrial waste. Agricultural processes as well as waste from fruit and vegetable processing industries generate large quantities of biomass which is generally dumped or wasted. These waste still contain a lot of nutrients in the form of cellulose, nitrogen and phosphorus compounds which still can be exploited for various purposes. Mushroom cultivation technology is a field of biotechnology which cultivates edible as well as medicinal mushrooms by fermentation process and making use of such organic biomass as a substrate. This method converts the agricultural biomass into nutritive biomass which can be utilized for consumption and dietary purposes. Biosensors for monitoring of environmental contamination. Biosensors are biosensing elements like enzymes, antibodies or biological cells based analytical devices used to detect presence of environmental pollutants. These sensing elements or cells are immobilized on the surface of the biosensor. The enzymatic system catalyzes a biochemical reaction converting the substrate to the product. The amount of product formed during the reaction is directly proportional to the electrical signal generated by a transducer. The output from the transducer is amplified, processed and recorded. Many biosensors has been developed for monitoring different kinds of environmental pollutants. Genetically modified yeast cells were used as whole cell biosensors to detect presence of estrogen molecules in polluted waters. Food biotechnology. The field of food biotechnology majorly deals with the use of microorganisms or enzymes produced by them for processing of inedible raw material into edible food products like bread, cheese, alcoholic beverages, curd, etc. Some of the modern biotechnological applications of food biotechnology will be discussed now. Cheese making. Cheese has been used as a food product since more than hundreds of years. Traditionally, cheese making process made use of calf rennet as a milk coagulant. However, with the increasing food demand in India, the available quantities of calf rennet were not enough. Additionally, because of religious beliefs in India, calf rennet was not preferred enzyme source. Hence, to produce large quantities of enzyme chymosin, biotechnological tools were used to clone and express the gene for calf chymosin enzyme into E. coli and other hosts. Vegetable and fruit juice. Many vegetable and fruit juice processing requires enzymes like pectinases and cellulases in order to increase overall productivity. Pectinases enzyme can be extracted from fungus Aspergillus niger. Cellulases can be extracted from many fungi and bacteria that exhibit cellulolytic activity. Alternatively, through genetic engineering, the genes for these enzymes can be cloned and overexpressed to obtain large quantities of the enzyme to be used for vegetable and fruit processing. Yeast strain used during beer making is selected on the basis of the flavor and aroma imparted by the strain during fermentation. Fermentation rate, ethanol tolerance, osmotolerance, oxygen requirement are the other parameters considered during the selection of the strain. Saccharomyces cerevisiae strain is commonly used for commercial beer production. Many desirable properties can be incorporated in the strain to produce a genetically modified organism. Recently, new brewing yeast strains have been produced with ability to ferment wider range of carbohydrates and produces beer with modified flavors. Industrial biotechnology. Industrial biotechnology is a field of biotechnology which deals with production of chemicals, materials and fuels by using renewable raw material as a substrate and living systems or enzymes extracted from them as catalysts for the bioconversion. Industrial biotechnology is gaining increasing importance because it has less energy consumption and hence lower production cost. Additionally, it also produces less greenhouse gases and thus is environmentally friendly. Lastly, it also enables synthesis of valuable industrial products by making use of biological systems of enzymes rather than chemicals, thus benefiting our economy and environment. Metabolic engineering is an important tool of industrial biotechnology during which genetic manipulations of the enzymatic or the regulatory pathways is done such that these changes favor enhanced expression and production of the product of interest. In the following slides, we will discuss about two examples of applications of industrial biotechnology. Lactic acid production. Lactic acid or 2-hydroxypropionic acid is an important chemical reagent commonly used for a number of purposes like as food preservative and decontaminant in meat processing, in detergent industry as a soap scum remover, in cosmetic industry as a chemical exfoliant and many more purposes. Traditionally, D and L-lactic acid was produced by Lactobacillus species during fermentation reaction. However, these bacteria have special growth requirements like amino acids and sugar cane or corn stip liquor. Hence, other bacterial systems like E. coli were modified through genetic engineering and were used for commercial production of D and L-lactic acid. In E. coli, TG114 strain was designed to ferment hexoses by knocking out the genes for competing pathways and deleting the methyl glyoxyl synthetase gene to produce optically pure D-lactate with high productivity. Biofuels. Decreasing fossil fuel resources like petroleum, increasing cost of harvesting, processing and distribution of fossil fuels and environmental pollution associated with these processes are the major challenges faced by the world. These concerns have prompted the research and development studies on use of renewable energy sources like bioethanol, biodiesel, etc. In chemical biodiesel production process, lipids or triacyl glycerols are esterified through trans-esterification reaction with short-chain alcohols like ethanol. Vegetable oil or animal fat are commonly used as substrate or lipids for the trans-esterification reaction. Researchers have engineered an E. coli strain by deleting the FADA gene encoding the acyl-CoA synthetase involved in fatty acid degradation. The recombinant E. coli strain was capable of producing 0.024 grams of fats per gram dry cell mass. Researchers are also screening and investigating new microbial strain with potential to produce lipids to be used as substrate for biodiesel production. Forensic science. Forensic science is a field which deals with detection and investigation of the crime based on the evidences found at the crime scene. Some forensic scientists travel to the crime site to collect the evidence themselves, while the others are involved in carrying out laboratory testing. DNA fingerprinting technique is commonly used to distinguish between different individuals by making use of DNA as a sample. Any two humans or individuals of the same species has majority of their DNA sequences in common, hence sequences that are variable must be analyzed. In the following section, we will discuss some of the biotechnological techniques used for forensic analysis. Figure 16. Restriction fragment length polymorphism. The term restriction fragment length polymorphism refers to the difference between two homologous DNA samples that produce fragments of differing sizes upon restriction digestion with a restriction endonuclease. This difference in size of the fragments produced can be because of a presence of additional recognition site or absence of a recognition site for a particular restriction enzyme in the location of the genome analyzed. The RFLP analysis involves processing of the sample to extract DNA, digestion of a DNA with a particular restriction enzyme, for example EcoR1, separation of the resulting fragments by agarose gel electrophoresis, Southern blotting to transfer the fragments onto a membrane, probing of the membrane with specific probes that have been proven in the past to be specific, heritable and suitable for RFLP analysis. On probing, a specific DNA binding pattern will be generated that is specific for each individual. Figure 17. PCR based analysis. An alternative method to RFLP analysis is a PCR based method that is used to amplify the variable number of tandem repeats. These are part of the DNA containing specific sequences duplicated in variable number of times in different individuals. For example, one VNTR D1S80 present in chromosome number 1 is repeated from 16 to 40 times in different individuals. An individual homozygous for the chromosome 1 will have equal number of repeats on both chromosomes and hence yield a PCR product of the same size, while an individual who is heterozygous for chromosome 1 will yield PCR products of two different lengths. Thus, both methods are useful for forensic analysis. However, the PCR based method is less time consuming and requires less sample. The major issue associated with the DNA typing methods is the amount of time required for the processing of large amount of samples. New instruments and methodologies are currently being developed with the aim of decreasing the time required for processing large amounts of samples. Ethics in biotechnology. Genetically modified plants and animals have many applications including increased productivity, enhanced nutritional content, etc. Genetically modified animals also have potential applications in medicine, whereby these organisms are used for animal studies. Different groups in the society have different views about biotechnology depending on their cultural backgrounds and previous experience. Many biotechnological studies make use of genetically modified animals for experimental model for studying human diseases, clinical trials, or drug testing. Many individuals, such as animal liberationists, are against the use of animals. Others support this since it may help us develop new human medicines. It may also help reduce the number of experimental animals used. The genetically modified plants also have many advantages. For example, golden rice is a good source of beta carotenes to satisfy the vitamin A requirements. Bt cotton is pest resistant and hence leads to increased productivity. Many groups support the use of transgenic plants because they have better quality and quantity. However, many groups oppose the use of transgenic plants because of the possible allergic reactions, as well as because of the possibility of gene transfer to the wild weeds, resulting in generation of super weeds. Limits of genetic engineering thus need to be established based on the public as well as expert opinions, especially for those genetically modified organisms that are intended to be released in the environment. Thus, in summary, in this lecture we have learnt about the concept of biotechnology, genetic engineering as an important tool in biotechnology, varied applications of biotechnology with examples, and ethical issues associated with biotechnology. Thank you. You can also find this lecture on the UGC ePathshala website.